Great, thank you. So today I'll be sharing some of the recent work we have been doing in the group in trying to better understand how cancer cells make decisions, specifically during the journey of metastasis. Now, cellular decision making is something which is being studied extensively, both experimentally and computationally, and of course in integrated effort in various different systems, in various different groups all around the world. And the central question that this community focuses on is how does one cell or a combination of cells in space and or time decide which of the finite discrete decisions to undergo, depending on a whole myriad of different biochemical and biophysical signals that it is exposed to, which of course vary as a space of function and time as well. Now, what we have seen over the past 3 or 4 decades since these observations have become much more precise is that even if you have an isogenic population, which means cells with the exact same genetic background, and you expose them to the same signal, you would still possibly see that cells respond differently. What causes this heterogeneity is something which I'll also talk about a little bit today. So this decision making at the end of the day is coordinated by different interconnected networks, an example of which in the form of a cartoon that you see here. So today I'll be focusing on metastasis, which is what the focus of our lab right now is, and which is where I started thinking about systems biology research during my PhD almost 10 years ago now. Metastasis, as many of you may know, still remains the major cause of all cancer-related deaths. There are no unique mutational signatures which have yet been identified for this process. So we cannot so far at least associate a specific set of mutations which can be associated by saying that these mutations, when enriched for or depleted for, enables cancer cells to move from one organ to another. Now, metastasis is clinically insuperable so far. There are no specific therapies against metastasis. But what excites me studying about this process the most is that it's highly inefficient. More than 99.9% of cells which actually leave the primary tumor to get onto this freeway of blood vessels spread all over the body are not able to metastasize, which means that they are not able to form tumors at other organs in the body. But what is so special about that minuscule percentage of cells that are able to achieve this humongous task is what we try to understand better. Now, during metastasis, the journey of a cell is highly challenging because the environment around that cell which has decided to lodge itself away from primary tumor or its home organ and is going on a journey to hopefully establish a colony at another organ. The environment around it is constantly changing. So, for instance, if it's in the bloodstream, there is varying levels of shear stress being applied. There are varying levels of oxygen and nutrient being available to the cell and so on. There are, of course, immune cells which are attacking these cancer cells so that they are not able to complete this process. And we have different therapies as well, which are trying to thwart the success of this process. So during to be able to pass through these multiple bottlenecks, what cancer cells need is a fast and reversible adaptation mode. And mutations, unfortunately, provide neither of them. Neither are mutations fast nor they are reversible. So reversible, we already know about that mutations can be passed on from generations. In terms of this speed or time scale of the process, if you think about the typical time scale in which a cancer cell stays in a bloodstream, it's of the order of 1 to 2 weeks, which allows for possibly 10 cell divisions. And we all know that the rate of error in copying of DNA is extremely low. And even if that error happens, what is the likelihood that it would happen in a specific gene which would actually provide an advantageous feed to the cancer cell? So therefore, mutations are not necessarily the way to think about it. They are a perfect way to think about cancer progression, primary tumor, and so on, but not necessarily the process of metastasis. So what has been identified in metastasis is the ability of cells to switch their phenotype or their behavior in a reversible manner and a fast manner in response to these diverse environmental conditions. And this is what is called as the phenomena of cellular plasticity, which can exist on multiple different axes. So these can be on the metabolic axis, drug resistance, their ability to migrate or settle down, and so on. And this has also been recently identified as one of the hallmarks of cancer in the year 2022, when the latest review on this theme was written. So the questions of our interest are, how do cancer cells switch among these different phenotypes in a reversible manner? How can they change their behavior along multiple axes? So once they're beginning to move, are they gaining drug resistance simultaneously? Are these networks coupled to each other? Are they also changing the way that they are going to metabolize the nutrients, and so on and so forth? And finally, once we know how these decisions are being made, can we alter those transition trajectories? So something similar to reprogramming or transdifferentiation kind of therapy, which has been beginning to be talked about. So most of our work has focused on this one of the most well-characterized axes of plasticity, called epithelial mesenchymal plasticity. What this means is that if you think about the most common solid tumors, such as breast cancer, prostate cancer, liver cancer, lung cancer, etc., those are all epithelial organs. So cells do not typically migrate or invade, rather they settle down in a specific 3D geometry and are adherent to their neighbors. So this is the typical epithelial behavior. On the other hand, we have mesenchymal cells in the body, which have the exact opposite set of properties, that they migrate and invade usually and do not typically adhere to their neighbors. So this transition, where epithelial cells, when they begin to metastasize, when they begin to leave the primary tumor, gain some of the properties of mesenchymal state, it is what is called as EMT, or epithelial to mesenchymal transition. Now, once they have reached the other side, they no longer need to migrate, invade. They actually need to settle down and reform the bonds with their neighbors, which they had earlier lost, and which is called as the exact opposite transition, called as MET, or mesenchymal to epithelial transition. Now, when we started thinking about EMT about 10 years ago, these schematics represent what the thought process about EMT in the context of cancer metastasis was at that point of time. So as you can see in all these schematics, EMT is, or MET is usually posited as a very binary process, that cells are either epithelial or they are mesenchymal. There were some reports about the so-called metastable phenotypes, but they were not really shown as the end point of a transition. The other inherent latent assumption in all these schematics is that EMT is always reversible. And if sort of we extend the argument further, one other assumption, which is again hidden behind cartoons, is that EMT is symmetric. So we talk about some of those aspects today. Are how valid are these assumptions when you actually start to look into the data more carefully? So the first question we asked was, is EMT binary? And at that point of time, what we did was identified a bunch of players which can drive EMT or MET as reported in multiple different cell lines, and how they connect to each other. And then can we represent those interactions through a set of differential equations whose dynamics we can then solve. So here in this schematic, what you see are transcription factors, or two families of transcription factors, which have been lumped to two nodes for the sake of simplicity as a first effort. So these transcription factors drive EMT. The higher the levels of these factors, the more mesenchymal a cell is. And on the other hand, you have two microRNAs, which drive MET, or the higher the levels of these microRNAs, the more epithelial a phenotype is. Now, the numbers that you see here represent some sort of semi-quantitative input-output relationship that can be gained from respective papers. You can convert this diagram into a set of differential equations and then solve for its dynamics. So what our model predicted when we look at the typical dynamics of the system. So here is a bifurcation diagram where x-axis tells you the external signal, the levels of snail which are driving EMT, and the y-axis tells you the extent of EMT-ness. The solid blue lines are stable states, and the dotted red lines are unstable states. So what we see here is that clearly there is an epithelial phenotype with very low levels of ZEB. There is a mesenchymal phenotype with very high levels of ZEB, but EMT is not binary. You can see a third state which has intermediate levels of ZEB, higher than epithelial but lower than mesenchymal. And this is what at that point of time called as a hybrid epithelial mesenchymal phenotype. Now that's one prediction of the model, that EMT is not binary. There are other predictions as well, one of which being talking about coexistence of phenotypes. So if you look at in this particular region of the diagram, it corresponds to only one phenotype here, which is epithelial. On the other extreme, you have again only one phenotype possible for a given value of snail, which is mesenchymal. But in between, you can have scenarios in which more than one phenotype can exist. And this is what refers to as non-genetic heterogeneity or phenotypic heterogeneity, that you have the same genetic background but different phenotypes. So these were the three predictions. EMT is not binary, there can be non-genetic heterogeneity, and because there are multiple states that coexist, they can switch back and forth under the influence of noise, for instance. So we collaborated with a set of experimental colleagues to test out these predictions, and here is some experimental validation. So here in these images, what you see are lung cancer cells stained for an epithelial and a mesenchymal marker called E-cadherin and vimentin, respectively, in green and red. So what you see is that most cells do co-express red and green, and they continue to do so over multiple passages in the lab. So it's not just a snapshot, it's possibly a stable state that can be maintained in vitro across multiple different cultures, passages. You can quantify this in much more detail. So there are, these were lung cancer cells. Now, then further looked at breast cancer, prostate cancer, another lung cancer, and colorectal cancer cells through this flow-based sorting for the same two markers that I was referring to. And you again see heterogeneity, cells present in all these different quadrants, despite having the same genetic background. Now, if you were to segregate these cells, these three different scenarios, which is what has been done in this experiment, you and culture them independently, what you observe is that without any induction as a function of time, cells can switch back and forth. So the population that started as 100% epithelial at the end of 2 weeks is no longer 100% epithelial because some cells have switched to being either hybrid or a mesenchymal state. So work done by us and various other experimental and clinical colleagues simultaneously in the past decade has now changed the thought process or the paradigm about EMT from thinking about it as a binary process to a process which has one or more of these intermediate states. And not only these states exist, but also they are crucial for metastasis, as you can see in this particular schematic. And also there have been other papers claiming all the way that hybrid states are essential to form tumors when it comes to metastasis. This is some clinical data where we see that the enrichment of these dual positive cells is seen in the most aggressive form of breast cancer called triple negative breast cancer. And there's further clinical data in these plots. The x-axis is time, the y-axis is likelihood of survival of a patient, whether it's overall or disease-free survival. And these two scenarios that you see represent whether a patient has a hybrid cell population or not, as gauged based on the pathology. And if the hybrid cells are as low as 2%, that dramatically decreases the likelihood of survival of this patient. So clearly it has a clinical role to play as well. Now, we were not the only ones making mathematical models of EMT. There were various other groups, and I'll quickly walk you through some of the work that our colleagues have done in the field. So there have been extensions of the networks that we looked at earlier. So these are still ODE-based models where you have specific kinetic parameters for which you look at the dynamics. Here is an example. We looked at ZEB, snail, MIR200 and MIR34, this network here. And in this particular example, more nodes have been added. So the dimensionality of the system is higher now, and that can possibly give rise to one more state as well. There have been much larger networks that colleagues have looked at, and because once you look at such larger dimensionality, you don't really have parameters available. people usually stick to doing a discrete model scenario. And when you do the discrete models or logical models, Boolean models, and then project them on a two-dimensional epithelial and mesenchymal plane, you again see that there are canonical epithelial and mesenchymal states, but there is a whole spectrum of hybrid states that have been reported. So despite these differences in modeling strategies and networks, there have been some consistent observations reported from the whole community of EMT modeling in the past 10 years, one of which is that EMT is not binary. This is an example I showed you earlier. Here is another example where a large network was modeled through differential equations, but instead of one particular kinetic parameter set, the authors took an ensemble of parameter sets and then looked at what the different steady states possible were. So here you can see that epithelial and mesenchymal are still the dominant phenotypes with one or more of these hybrid states. There have been other questions that we and others have been asking in terms of, can we look at the network topology itself and comment upon which states are more likely to exist in terms of epithelial, mesenchymal, and hybrid, or combination of them. So what we'll talk about today are some questions that we have been thinking for almost past 3 years. So one observation has been that no matter which of these larger networks you look at, they have these common players that we started with. They are all extensions and built upon. They have that core network that we started with. So we have been thinking, what happens to the dynamics of this motif alone when embedded in much larger network? Does it continue to show the behavior it did in our earlier scenarios? And the other question we have been asking is that now if we look at multiple such EMT regulatory networks that different groups have identified, developed, and modeled, are there some common structures that you can see in those different groups? So the work I'll be talking about now is led by my PhD students Kishore and Pradyumna, with excellent contributions coming from other group members, Achyuta, who's also a PhD student, and Aditi, Varun, and Archana, or undergraduate students. So toggle switch or double inhibition feedback loop is something that many of you have already heard about. This was also one of the first synthetic biology circuits that was constructed in E. coli. So the typical dynamics of this networks looks like the following. Here is a graphical representation. So now if you look at how the levels of A change as a function of B, because A is repressing A, so the higher the levels of B, the lower the levels of A, which is what this curve represents. And if you look at the other behavior, which is how the levels of A are changing, how the levels of B are changing as a function of A. This is how the other curve looks like. In technical terms, these are called null clines. You look at the intersections of these curves and then figure out which of them are stable and which of them are unstable. You can do the typical stability analysis where you perturb the system and see whether the system comes back or the system actually goes to another state. So when you do that, what we find is that this system has two stable states, A high, B low, or A low, B high. In colloquial terms, this is like two people who hate each other and do not want to go to the same event. So either A ends up going and B is absent, or A is absent and B is present. Okay. So this is the typical property of bistability. These are typical equations which you would often see in mathematical models representing this kind of dynamics. So each variable has a production, has some regulation through B, and has some first order degradation. The reason why I'm focusing on this structure is because this is seen in multiple different scenarios when it comes to cell fate decisions. So these are examples from embryonic development. Whenever there is a bifurcation that comes in terms of cell fate, these kinds of structures show up in a much higher frequency than just a random structure would. So for instance, if a pancreatic progenitor has to decide whether to become exocrine or endocrine, these two molecules are seen to be mutually inhibiting each other. And this makes sense because when you have to take a decision, you have to stick to that decision, largely speaking. So one lineage has to be strictly on and the other lineage has to be largely off, which is what this toggle switch typically allows. So now, if we look at the dynamics of this toggle switch over a range of parameter sets, so we start with the typical ODEs that are shown here, generate randomly, say, 10,000 parameter sets, where the parameter ranges are defined based on biologically relevant values, simulate the ODEs for multiple initial conditions, take all the solutions together and see what different possible steady states exist. So when we do that for a simple toggle switch, as you would expect, it leads to these other density plots, typically a state where B is high and A is low, or the other extreme where A is high and B is low. You can also do a scatter plot, calculate the correlation coefficient, because these are all continuous ODE-based simulations. And what we see here is that A and B are strongly negatively correlated as expected. What this entity shows here is the bimodality coefficient, which ranges on a scale of 0 to 1. The higher the value, the more bimodal a distribution is. Now, this is standalone. What happens if you embed this in much larger network? So this is what we did. We generated multiple different networks, let's say of different sizes, so say of 20 nodes and of varying different number of edges. So if you're talking about this particular network, 20 nodes and 120 edges, and we generated 100 such random networks, which are connected. The nodes are connected in different forms. And in each of those 100 networks, we embedded a toggle switch and asked, how does the dynamics of a toggle switch change? So we are talking about 1200 networks here, 100 for each of the 12 cells that you see, 4 different sizes and 3 different densities. And we repeat the process and ask, does a toggle switch stay a toggle switch, or when does it begin to lose its properties? So here is the correlation coefficient, which, as you saw in the standalone dynamics, was somewhere around negative 0.8. So it's lying somewhere here. Now, in this case, because we have two independent variables, density and size, we first group the networks here based on density, and then see whether by changing the size, what is the distribution of coefficient correlation between AB in the pool of 100 networks that we chose for each such combination. So what we see is that if you fix the density and vary the size, there's not much change. But if you do the opposite, which is that if you fix the size and vary the density, then you begin to see significant changes. And the trend is also interesting because as you increase the density from e's to 2n, which means twice the number of edges as nodes or 6 times the number of edges as nodes, what you see is that correlation coefficient begins to decrease and go towards 0, which means that the antagonism between A and B, or the two nodes in a toggle switch, is slowly weakening. Similar trends can be measured by other entities. Bimodality coefficient, this should decrease because higher the value, the more bimodal it is. So for a typical standalone case, this was around 0.8, and it goes all the way to 0.2 approximately. F1 is the frequency of states which are basically in second or fourth quadrant. So A high, B low, or A low, B high, because there are also some states which are both low or both high, depending on the parameter case. So again, this value decreases, which means that the properties are slowly decaying as the density increases. So next we wanted to see, is the global density more important or local density? So here is an example. We take all the 100 networks and then club them based on the in degree of A, which means the number of edges coming onto A, or in degree of B. What you see is that the fastest decrease happens along the diagonal, which means that if both in degrees of A and B are increasing, the network is getting more and more densely embedded, then is when you begin to lose the properties. So here are some examples. So here this is standalone. There's in degrees A and B, which means they are just inhibiting each other. There is no other connection. So F1, which means the sum of these two numbers, is approximately 90%. Correlation coefficient is also approximately negative 0.8, and so on. And the distributions are reasonably bimodal. When you add many more edges on B, you see that the properties of B begin to lose, but the properties of A are still maintained. You can still see some bimodality here. When you have both of them as high, bimodality is gone on both the fronts. F1 decreases and correlation coefficient also decreases. So what we see here is that if you have higher number of edges, and that number is somewhere around 4 to 5 on each node at all times, as long as you don't have 4 to 5 edges coming onto each node in a network, a toggle switch largely stays a toggle switch. The properties are decaying, of course, but once you cross this threshold, the decay is even faster and more abrupt. Then we looked at what happens if you add self-activation or self-inhibition. So as expected, we did see that when you add self-inhibition, you are actually weakening the networks even further, but self-activation, you tend to preserve the behavior. And similar results we obtained for toggle triad, which means when there are 3 nodes inhibiting each other. So there's a general observation that we had was that it's the high local density, particularly irrespective of the sign, whether it's activation or inhibition, around a toggle switch, and self-inhibition a toggle switch, which actually disrupts these functions. So into networks and observe that the mir200 ZEB feedback loop that I was talking to you about does not really have more than 5 edges at the time. So which means that even if they're embedded in larger networks, they still retain their properties, largely speaking. So that brings us to the second part. Are there any common topological features in EMT networks, which different groups have constructed over the past couple of years? Now, in each of these networks constructed by a different group that you see here, what we have done is in this representation, mark each of the node or each of the gene in 3 different colors. Red means it's an epithelial node. We know in the literature that if the higher the levels of this gene or this node, the more epithelial a cell is. Blue is mesenchymal. And gray are ones which might have an epithelial or mesenchymal property, but as far as the network is concerned, it doesn't form any feedback loops. It's just either an input or an output. In terms of edges, green edges show activation and red ones show inhibition. Now, if you look at this visually, these networks, you begin to see a pattern, which is what I'll talk about in more detail in a minute. But I sensitize you towards that, even looking at this in this visual representation. So these all mesenchymal players, as you might notice, are activating each other. They're not repressing each other. And because these are microRNAs, they can't really directly activate each other. So you don't really see activation within the epithelial set of nodes. But the other observation is that any node going from any edge going from one epithelial to another mesenchymal is inhibitory in nature, whether it goes from epi to mes or it goes from mes to epi. So we wanted to better understand this and see can we quantify this behavior in more detail and what implications it has in terms of dynamics of the network. Now, again, these are larger networks. We have no clue about kinetic parameters. So we take a typical Boolean modeling approach. What that means here is we are taking a simple majority rule. If the number of activation coming onto an edge is more than the number of inhibition, you turn the switch, you turn the node on. So each node can have only 2 values, on or off, +1 or -1. If the number of inhibitions is more, then is when you turn the node to be off. And if they are equal, you don't change the properties. And you basically look at the steady states that are being resultant from these networks. Okay, so if you look at this example, we have 2 to the power 22 unique initial conditions possible because each node can be on or off. So we start from a large ensemble of initial conditions of the order of 10 to the power 6 and see what different trajectories that this network shows and what steady states it evolves into. So what you see here for this network is that there are epithelial phenotypes, which means that the levels of all these nodes is high and the levels of all these nodes is low. And of course, there are the mesenchymal states, which means that the levels of all the epithelial ones is low and all the mesenchymal ones is high. So that's clear. In addition, you see many of these hybrid states also show up. So then we quantify this further. What is the frequency of these states in all the 5 networks that I showed you? And we see a pattern, with the exception of this particular network, the epithelial and mesenchymal phenotypes actually dominate the steady state behavior as compared to the hybrid ones. So the hybrid ones are not as frequent. So then we asked whether this property was specific to these 5 networks. The way we asked that question was, we would take a network and then shuffle the edges, just as one would shuffle a deck of cards, and ask whether these properties are maintained or are they lost. So for a given network that you see here, we did this shuffling or randomization 1,000 different times. And for each of those 1,000 non-biological randomized hypothetical networks, we quantified how many steady states the system has. And this is the histogram showing that. So what you see here is that this is where the wild type is, the red line, which has a small number of phenotypes. But if you randomize the networks, you get many, many more number of phenotypes. The importance of this I'll just talk about in a minute. So that was one aspect, that there are some properties specific to the biological network structures. The other property we looked at was something called coherence. What you see here is that if you start from a state and perturb a state, think of it as some random noise. What we do here exactly is flip one node, any one node, out of n nodes which that network or that state has, and then quantify how many times does it come back and how many times does it go away to other states. So it's like doing local stability analysis, but once in every dimension that the system represents. When we plot a distribution of those values, so this coherence can lie on a range of 0 to 1. The higher the value, the more coherent, the more stable that state is. So you can clearly see across all 5 networks, there's a bimodal distribution. There's one set of states with low coherence values, another set of states with reasonably high coherence values. We quantify another property, which is we call, in fact, it's taken from a PRL paper that was published almost a year ago, called frustration, which tells you how strongly a state obeys a topology. So, for example, if A activates B and both of them are on, it's not frustrated. But if A is on and B is off, it's a frustrated state. So you can quantify similarly how many edges do not obey the topology in a given state, and you can quantify that, and again you see a bimodal distribution in these scenarios. Now when you look at both coherence and frustration together across all the 5 networks, we see a very clear behavior. The higher the coherence, the less the frustration is. And now on top of that, you overlay the frequency, and the picture becomes very clear. There's one set of states which are either epithelial or mesenchymal, which have very low frustration, very high coherence, and high frequency. So these are the dominant states across all 5 networks. So it doesn't depend on just the choice of network. And then there is this other set of states which have high frustration, low frequency, and low coherence. And this is what sort of beautifully explains this kind of experimental data which was coming, that hybrid states are easiest to switch out of, but epithelial and mesenchymal are more stable. So then we wanted to understand, why are they more frustrated? They're more frustrated, it's easier to switch, but why? Is there a reason behind that? So this is when we begin to quantify the kind of visual appreciation of networks that I was talking to you earlier about. So what we do is convert this network into a matrix. Now here, row is the source, column is the target. Red means there is an activation edge going from this particular node to this particular node. Blue means the inhibition, and white means there is no regulation. So in this 22 cross 22 matrix, there are only 82 edges. So it's a sparse matrix, as you can clearly see. Now, this is about direct edges, but as we know that a signal can propagate through a longer path as well. So let's say if A activates B directly and A inhibits C, which activates B. So A is also inhibiting B indirectly. So what we have done here is taken such direct and indirect paths going between any two edges and collapse them to form one single edge. So in other words, we have multiplied the matrices such that we can get the impact of a longer path rather than just the direct path. And this is what we call as an influence matrix. This is not just adjacency, it's about how strongly one node can influence any other node. And this is where this scenario becomes crystal clear. All epithelial nodes effectively inhibit all mesenchymal nodes. All epithelial nodes activate all epithelial nodes. All mesenchymal activate members of the same family, and all mesenchymal inhibit members of the other family. So at the end of the day, this seemingly complex network is a toggle switch where entities are not just two molecules, but two teams of molecules. Not every team player might be equally hating every other team player on the other side, but effectively they're doing the same thing. Okay. Then this is what we observe through simulations as well. So this identification of teams requires no simulations. All you need to do is take a look at the network, construct adjacency matrix, and multiply that matrix depending on how much path length you want to consider. But it can still give you the most dominant states present from a network. Now, again, if you shuffle the network, you see that this property of teams is completely lost. So we quantify team strength again on a scale of 0 to 1. Higher the team strength means, the stronger these antagonistic patterns here are. So for all the random networks, you can see that the mean of this distribution is somewhere around 0.3, whereas the wild type lies somewhere around 0.7. No matter what the absolute value of team strength is, we were able to show that using this influence matrix alone, which required no simulations, we were able to predict the most dominant phenotype with an error within an error range of 2%. So that saves you a huge amount of computational cost. Okay, so teams exist, so what? How does it change the properties of EMT or MET? So here is an experiment, a perturbation experiment, where you basically start from either an epithelial, mesenchymal, or hybrid state, and then you perturb these nodes that are written below, and you see whether the system comes back or does it stay there. When you start from hybrid states, they most likely go away to another state, but when you start from epithelial or mesenchymal, they stay there. You can quantify this further by doing more than one perturbation, which is what you can see here. So if you are starting from an epithelial or a mesenchymal state and you want to reach the other state, it takes approximately 30 to 40% of nodes to be flipped, on ones to be turned off and off ones to be turned on. So it's a large enough perturbation. But if you start from hybrid states, as long as you have flipped 10% or 5% of the nodes, you have actually pushed the cell out of a hybrid phenotype. So it's much easier to switch from a hybrid state to anywhere else, but the attractors of epithelial and mesenchymal phenotypes are much deeper in general. So that's the sort of summary of this part. And again, the strong teams is something that is a property of wild type networks and not really seen in randomized non-biological networks that we looked at. So then we started thinking, and this is very much work in progress, do teams exist in other networks, and why do teams exist at all? So here is another example of phenotypic plasticity that we looked at in another highly aggressive cancer called small cell lung cancer, less than 7% rate of survival, 5-year survival. We had a network already which was published in the literature. We simulated the dynamics of this network. This work was led by Lakshya, Uday, and Kishore. And what we found was that, again, despite a very large network, it predominantly had only 4 states. And if you randomize the network, the number of states that you see from all these 1,000 random networks are very, very different than what the wild type network gives you. So this gives you somewhere of the order of million states and so on. So again, this is a property specifically of this structure. We construct the influence matrix, we see the same behavior. 22 molecules effectively activate each other, 11 molecules activate each other, and then there is this mutual antagonism. So we can actually reduce that very complex network all the way to a very simple network of 4 nodes because of such huge amount of redundancy that is present in the network. You look at the simulation results, do pairwise correlation, you can see these teams. And when you look at experimental data from cell lines, you again see this very consistent behavior, suggesting that the network that we have is able to recapitulate the different experimentally observed phenotypes. We also saw these 4 different phenotypes based on the specific molecules, which were identified based on the composition of these groups. Another example is work led by Malvika in the group, looking at melanoma, another cancer which is not an epithelial cancer, so doesn't follow EMT paradigm, just as the small cell lung cancer also doesn't follow it. Again, we identified a network, modeled the dynamics of this. We see this behavior of teams coming up, one set of molecules strongly positively correlated with each other in terms of simulations, and so does the other one, and there is mutual antagonism. This is what we see experimentally as well in specific data sets. And here we talked about additional microstates that can emerge from this proliferative and invasive states. So then the question comes, why do teams exist? So one obvious answer is to build some redundancy into the network. This is some analysis we have done, that in the small cell lung cancer network that I showed you, if you were to delete one edge at a time, this is like crispering out an edge, 345 edges when deleted one at a time have no change in phenotypic distribution out of 357. That tells you the extent of robustness, and of course it leads to a limited number of steady states, which we already saw. One other direction which we are currently thinking about is that teams might also help connect multiple axes of plasticity. So an example here is EMT and drug resistance. So here this is an estrogen receptor positive breast cancer, which is work led by Sarthak. Here is a network we have which has the EMT players, and it also has the drug resistance players, the tamoxifen, which is the first targeted therapy and the most common given therapy to estrogen receptor positive patients. So we saw a correlation that the higher the EMT, the more likely the cell is to be resistant. Now, we then checked whether there was a causative connection. So our model predicted that when cells undergo EMT, you drive resistance to tamoxifen. And then we looked at transcriptomic data, and we did see that in cells undergoing EMT, you saw a repression of estrogen receptor signaling pathway, which is what is responsible as a major factor driving resistance to tamoxifen. And the other way around as well. If you have gained drug resistance, that can drive EMT as well, which is what the model predicted. And in this transcriptomic data, we saw that when you silence estrogen receptor, therefore driving tamoxifen resistance, you see an enrichment of all these mesenchymal players. So what is happening is a bidirectional knob. When cells are beginning to move, they are carrying the property of drug resistance with them. And the drug which is given in the clinic has a potential side effect of accelerating metastasis. So what we are proposing now, and this is being experimentally tested right now in collaborators group, is that give tamoxifen together with EMT inhibitors, because then you can counteract the side effect of tamoxifen, which is driving more metastasis. The similar behavior we have seen in prostate cancer, where epithelial mesenchymal transition and androgen resistance is coupled. This is work led by 2 undergraduate students in the lab, Rashi and Abhibsha currently. And again, we see the behavior of teams, as you can clearly see in this scenario, the 2 teams and the mutual antagonism, as well as an experimental data. So I'll just end here. In multiple different networks that we have seen, we do see that these networks have the property of multi-stability, which can allow for cells to switch back and forth under noise or under various other perturbations, including the perturbation given by the drug. One of the reasons why these complex networks lead to limited phenotypic repertoire is because there are existence of teams which are behaving as a canalizing principle, reducing the number of total phenotypes. And we are trying to use these networks to better understand how cancer cells respond to drug and develop alternate trajectories of reversible reversible drug resistance as well. Based on that, which we can then identify combinations and sequence of therapies. So I know it's a long claim, but we think that teams are behaving in a very similar manner to energy minimization, um, which is why you see that there are only a few limited structures of a given nucleotide sequence possible, a protein in the context of protein folding. Our teams playing a similar role when it comes to phenotypic possibilities or steady state or stable states that was possible. With that, I would like to thank the fantastic set of people and collaborators and colleagues who have had the fortune to work with. And last but not at all the least, all my team members who come from diverse backgrounds, enriching each other and trying to address these questions about how cells switch back and forth and how can we use this better understanding of the dynamics to tackle metastasis. Thank you very much once again for this forum, and I'll be very happy to take questions and suggestions. Thank you.
Right. Thank you, Mohit. That was an excellent talk. We do have some questions in the Q&A tab. So let's start with Vipul's question. Vipul is joining us from the UK, and he asks whether the lifespan of hybrid cell is same as epithelial and mesenchymal cell.
Yeah, that's a great question, Vipul. So if by lifespan we are referring to the stability in terms of how long a phenotype can be maintained, then we do see that hybrid ones switch faster as compared to epithelial and mesenchymal. If we are referring to proliferation rate or viability, then these are comparable. Mesenchymal has a slightly low proliferation rate as compared to epithelial and hybrid, but viability, at least so far, I have not seen literature talking about the differences in there.
Yeah, another question from Vipul again was under different networks, are the strengths of A inhibiting B and vice versa the same?
Yeah, that's another great question. So we see this behavior of teams, and we do see scenarios, at least in these networks, that it's comparable. The inhibition coming from team A to team B and vice versa. We are now constructing artificial networks where we are varying these inhibition strengths and then seeing whether it leads to a change in the phenotypic landscape such that where the inhibition is stronger, is that an easier state to come out of or a less frequent state to obtain and so on. So that analysis is still under progress. Another thing we are doing right now, so far everything is two teams, but in real networks there can be three teams or four teams. We don't know that yet. Those are the networks which we are artificially constructing hypothetical networks and then seeing the properties in terms of both dynamics and topology.
Thank you, Mohit. I had one question. So when you talked about shuffling the network, um, how do you make sure that the properties stay consistent? So the inhibition and the respective activation of these nodes are consistent.
Right. So what we make sure is that, of course, the total number of nodes and edges in the network is not changing. And also the total number of incoming edges on any node is also not changing. The outgoing degree is also not changing. What is changing is, let's say, if a node has 5 edges coming onto it. Now, in the wild type network, 5 out of those 5, 4 might be inhibitory and 1 might be activatory. But in a random network, that distribution can be 2, 3, 3, 2, 1, 4. So that distribution is changing in terms of how many edges are activating versus inhibiting, but the total number of inputs is not changing. And the total number of inhibitory and activatory edges in the entire network is also not changing. So what you're doing is basically swapping an activation with an inhibition or vice versa. And there are many ways to do it. So when you do it in many different scenarios, you actually lose the properties, largely speaking.
See, okay, yeah, thank you. And another question based on one of your very last slides regarding introducing MET inducers. So, is there also an importance of like time in this whole dynamics, like when it should be done in terms of like the whole transition of like, okay, now the EMT transition is happening, as well as the right time to give these inhibitors?
Definitely. That is something which we still do not completely understand, whether this should be sequential therapy or combination therapy, and if sequential, then which should be given first, and so on. Those are open questions. What we are saying so far is that there is some merit in looking at the side effect of the drug itself and identifying strategies how to counteract that side effect.
Yeah, no, that makes sense. Right. Well, thank you so much, Mohit, for presenting your work and sharing your work with us and answering all the questions. Really appreciate it. I don't see any more questions in the Q&A tab, but feel free to reach out to our speaker. I'm assuming that's all right, Mohit. And
Definitely.
yeah, the participants can reach out to you if they have any further questions. And with that, yeah, I would like to thank you again. And
Thank you so much.
yeah, Seth, let me know if there's anything, or we can wrap up.
No, that is everything. We do have one new question though.
Oh, okay.
They say they
Oh,
might have missed this. An interesting idea of explaining different phenotypes through Teams metric. Is there any biological relevance of TEMS?
Yes, that's a question which we are also currently thinking about. We have looked at two different developmental scenarios as well, where we do see the evidence of teams. One of which is in gonadal cell fate determination, when a cell has to decide to become between an ovary and a testis, which cell fate to choose. We do see very strong evidence of teams there. And another instance we have seen is in Th1, Th2, Th17 CD4 positive T cell differentiation, where we see that the so-called master regulators of these phenotypes actually repress each other in a reasonably strong manner, forming teams. And those are the two developmental scenarios. Most of our other examples so far have been from cancer, but we are very interested in looking at how similar or different teams in development versus teams in cancer are.